Welcome. In this section, we will talk about patterns of inheritance. To begin with, in January 2020, it was recorded that there are over 7.8 billion people all over the world with diversity of traits in skin color, hair color, having freckles or not having freckles, clipped chin or smooth chin, height in terms of being tall or short, among other traits. The heredity unit that contains information for a specific trait is referred to as a gene. During crossing, about 20,000 to 25,000 genes are carried from the father and the mother to the offspring. The genetic information are stored on the nucleus of the living cell, and within the nucleus there are the chromosomes which contains DNA, also referred to as deoxyribonucleic acid, the molecule that carries the genetic information. So, chromosomes exist in matching pairs, with one chromosome of each pair being inherited from each biological parent. Of the 23 pairs of chromosomes in humans, 22 pairs are the same, regardless of whether someone is male or female. And these chromosomes are called the autosomes. The remaining pair of chromosome is the sex chromosome, and that determines the sex of the individual. For the female genotype for the sex chromosome, it is represented by XX, and the male genotype for the sex chromosome is represented by XY. Alleles are the alternative forms of a gene. For example, hair color can exist as a black hair color or a blonde hair color. And another example is the eye color brown, green, or blue eye color. So the alternative forms of a gene is referred to as alleles. Now, Gregor Mendel, who is also known as the father of genetics, through his work on pea plants, discovered the fundamental laws of inheritance, and he crossbred over 30,000 pea plants and observed how changes in traits existed from each generation onto another. The first law that he came up with was the law of segregation, and in this law, he explained that for every gene, there is an alternative form, and these alternative forms are referred to as the alleles. During meiosis, these alleles segregate, and the offspring inherits one of the genetic allele from each parent when the sex cells unite in fertilization. And that is what creates diversity among the offspring. In his second law of independent assortment, he explains that the segregation of the alleles during meiosis is independently governed. So the genes for different traits are sorted separately from one another, so that the inheritance of one trait is not dependent on the inheritance of another. Mendel also saw in his findings that an organism with alternate forms of a gene will completely express the form that is dominant in the presence of the recessive trait. So after he crossed a green and a yellow, the produce was green, which indicated that the green allele was the dominant allele. Let's define some more terminologies. So for the crossing, if an individual has two identical alleles for a certain trait, then it is called homozygous for that trait. And if the organism has two dominant alleles, for example, capital B, it is called homozygous dominant. Likewise, if the organism has two recessive alleles, small b, it is called homozygous recessive. And an organism that has one of each type of allele, capital B, small b, will be called heterozygous. Also, the genetic makeup of a trait, that is the alleles that an individual has, is called the genotype. And the physical expression of the genotype of a trait in its environment is called the phenotype. So the Punnett Square is a square diagram that is mostly used to predict the genotype of a particular cross, and it was named after Reginald Punnett. You start by determining the genotype of the parents. For our example, each parent has a heterozygous trait, which is expressed as capital T and small t. So you put one for the female and one for the male. The genotypes for female parents are by convention listed across the top of the Punnett square, whilst the genotype of the male parents are usually also listed down the left side. So inside the square, you then complete the matrix by listing each possible pair of alleles that is created by the cross of the two parents in the Punnett square. As shown in this Punnett diagram, one of the offspring from this cross is expected to be homozygous dominant, capital T, capital T. Two offspring are expected to be heterozygous, capital T, small t, and one offspring will be homozygous recessive, small t, small t. Hence, the genotypic ratio will be 1:2:1. One homozygous dominant, capital T, is to two heterozygous, capital T, small t, and one homozygous recessive, small t, small t. Note that only the offspring that receive a recessive allele from both parents, small t, among the four offspring will have the recessive trait. Hence, the phenotypic ratio will be 3:1. To do a dihybrid cross, which involves two traits, you first determine all possible combinations of alleles in the gametes for each parent, and then you do the crossing like the monohybrid style, so vertical traits versus the horizontal pair, and then you place each of them in the matrices and determine the phenotypic and the genotypic ratio, just like the monohybrid cross. Co-dominance is a form of inheritance in which both alleles are equally expressed in the phenotype, and how it differs from incomplete dominance is the fact that for the incomplete dominance, the mixture of the alleles in the genotype is seen in the phenotype. For genes that are carried by either sex chromosomes are said to be sex-linked inheritance. Examples include color blindness, hemophilia, and Huntington's diseases. It is usually males who are affected because they have a single copy of the X chromosome that carries the mutation. And in females, the effects of the mutation can be masked by the second healthy copy of the X chromosome. So, in our example, from crossing a carrier female and a healthy male, you end up getting one normal female, one carrier female, a normal male, and a colorblind male. So the recessive sex-linked conditions often occur in males because males have only one X chromosome, and a single recessive gene on that X chromosome will cause the disease. And that brings us to the end of this lecture. Hope you enjoyed it and see you all next time.